is an annotation pipeline, and annotates based on genomic elements, for example, known genes, location relative to gene, protein domain, and prediction impact, coding impact, nucleotide conservation, microRNA impact, linking elements, molecular function, biological pathways, prior knowledge, phenotype association, GWAS catalog, allele frequencies, EQTL databases, and disease ontology, and many other databases, and categorizes variants based of ACMG category 1 to 5, 1 being pathogenic, 5 being benign, and we call it advisor score. And I will show you a real case study how you can use SG Advisor. I will base this off of idiom study, patient number 9. Idiom study is a study happening here at Scripps, idiopathic diseases of men research study with 6 causes of rare diseases. For patient 9, it was a 9-year-old female, lifelong history of hypotonia, development delay, cognitive impairment, intermittent agitation. MRI showed subtle asymmetric volume loss in left hippocampus. Motor milestones were delayed. Onset of generalized tonic-clonic seizures was at 4.75 years of age. We did whole genome sequencing and exome sequencing. For whole exome sequencing variant summary, we have over 101,000 SNPs and indels, 18,475 are rare variants with allele frequency less than 1%, 1,053 non-synonymous truncating or splice site variants, 2 de novo inheritance variants, and 3 homozygous or compound heterozygous variants. To annotate the variant file, you can go on genomics.scripps.edu/advisor, and you can, we accept VCF files, complete genomics files, bed delimited file format. And to upload the file, we accept registered upload. You can create an account, and accept unregistered uploads as well. I already have an account. I log in, I go to upload file, choose file to upload, and I upload a VCF file. Once the file is annotated, you get an email and you can download the results from the results page. So idiom 9, I download the results. Also, since the summary files is quite big, I can get for whole genome sequencing, it can be up to 3 gigabytes file. It's hard for a scientist that doesn't know programming to analyze it. So we created, under tools, we created the SG Advisor UI, which is a user interface for desktop computers that works on Linux, Unix, Mac computers, and doesn't require any programming or command line knowledge to use it. So you can download the UI from here, and it's downloading. Once the UI is downloaded, you double click on the jar file. Also, depending on the file size, you can change. So you upload, to open the file, desktop, idiom 9, summary. It might take from a few seconds to up to a minute. So this is the tabulated view. You can see all of the annotations from variant gene annotation, gene type, non-coding RNA location, downstream, upstream. Further down, you can see the conserved genes, allele frequency, 1,000 genomes, elderly allele frequency, EQTL, epithelial genes, microRNA. Also, you can read about all of the different annotation on our website. To read about annotation, you can go on result description, which describes what each annotation means. And at the end, you have the advisor score. You can sort the file. We have 13 custom filters. For example, you can filter by coding variants with frequency, which you put the 1000 genome frequency. You can choose. You can filter by chromosome position, so if you know the position of interest. And one interesting filter would be known disease, which one value is in advisor score columns. And you have 25 variants of category 1 advisor score, meaning pathogenic variants. However, since this is a quartet, meaning mother, father, and proband, they affected the individual and the sister, you will have to load and compare the genotypes, so load and compare VCF files to load the genotypes and see if any of those category 1 variants are homozygous or de novo. So load VCF file. Once again, you can see. So the genotypes are loaded up front. This is the proband. You will have to do the known disease filter again. And so these are all of the category 1 variants. However, when you look up front, none of the variants are homozygous or de novo in proband. Meaning none of those variants are probably not the causative variants. So to go back to the original data or to move in between filters, you can press on this one. So we go back to the original data and we can filter again on, for example, idiom filter might be helpful. Idiom are all de novo variants, homozygous in child only or compound heterozygous. Idiom filter, father is unaffected, mother is unaffected, proband is affected, and sister is unaffected. Filter. And just a few variants. There are some homozygous variants. De novo variant. Another one, de novo variant. And this variant in gene KSNP1. After literature search, my PI, Dr. Torkamani, found out is the pathogenic variant. So from 101,000 variants, we reduced it to just a few, and this variant, if you look down, it doesn't have an advisor score category because it was not found previously to be causative of this disease. However, it is probably damaging according to polyphen. After we found this variant in this person, 2 other individuals were found to have variants in the same gene, and the two other individuals had epileptic encephalopathy. And this was recently published in a paper, de novo KS NB1 mutation in epileptic encephalopathy, all the Turkamani being right here. And thank you to Dr. Turkamani, Philip Fan, and Bill Shipman. Thank you.